Hello, my name is Andres Pineda, and together with Maria Jose, I explore whether Vicalin can make lung cancer cells self-destruct by shutting down the glutamine mTOR pathway.
Introduction. Bakelin, a natural plant flavonoid with antioxidant and anti-cancer effects. Apoptosis. Programmed cells death, a natural way for the body to remove damaged cells. mTOR, a key protein that controls cell growth and metabolism. Lung cancer, a disease where abnormal cells grow uncontrollably in the lungs.
Non-small cell lung cancer cells are addicted to glutamine metabolism. They bring glutamine and leucine inside throat through transporter ACST2 and LAT1. One inside, glutamine is converted by GLS1 to feed the energy cycle, while leucine helps to turn on mTOR, which drives growth and blocks cells death. Vicalin shuts down the system, it lowers ACT2, LAT1, and GLS1. So glutamine use drops, mTOR weakens, and cells stop proliferation and start going into apoptosis.
Objective is to investigate if Bakelin induces apoptosis in lung cancer cells by modulating in the glutamine mTOR metabolic pathway.
The fundament about the Western blot detects specific proteins. It separates proteins by gel electrophoresis and then uses antibodies to identify the protein of the interest. Proposed in this article, in this study, Western blot was used to measure proteins linked to apoptosis, for example, BAX, BCL2, caspase, and to the glutamine mTOR pathway, for example, ACD2, LAT1, GLS1, and mTOR. It shows if Vicalin changes their expression in lung cancer cells. The fundament transfection introduced foreign DNA into cells, often use plasmids to make them overexpress or silence a target protein. The purpose in this article, or in this study, is the cells were transfected with a plasmid to overexpress ACC2 and LAT1. The goal was to test if restoring these transporters could rescue mTOR activity and block the apoptosis even in the presence of Vicalin.
PCR. Fundament QRT-PCR measure gene expression by converting mRNA into cDNA and amplifying. Purpose in this article. Use it to test in branchial reuse ASCT and ALAT1 mRNA levels, confirming these transfers and downregulated. Apoptosis. Fundament. Apoptosis assays detect if cells are undergoing programmed cells death, often by flow cytometry with annexion staining. Purpose in this article show that Bakelin increases apoptotic lung cancer cells, confirming its pro-apoptotic effect.
In both H1299 and A549 lung cancer cells, the control plates are full of colonies, and cisplatins reduce them partially. As Vicalin concentration increases, the colonies disappear almost completely. This shows that Vicalin has a strong anti-proliferative effect. At higher doses, cancer cells lose their ability to grow and form colonies, which confirms its tumor suppressing activity. Here we use Western blot, which detects proteins through antibodies. The goal was to check if Vicalin changes apoptosis-related proteins in lung cancer cells. As the Vicalin doses increase, BAX and cleaved caspase 9, BAX and cleaved caspase 9 and 3 go up, while BCL2 goes down. In both H1299 and A549, these two lung cancer cells, this pattern clearly shows the Vicalin pushed the cells toward apoptosis. Pro-apoptotic proteins increase and the anti-apoptotic proteins decrease.
Results figure 6, panel ABC. In the panel A, direct interaction of Bakelin with ASCT2, LT1 and GLC1 is observed. In the panel B, a dose-dependent decrease is ASCT2, LT1 and GLC1 is observed in H1299 and A549 cells treated to the Bakelin compared to the control group. There is also a significant reduction in total of phosphorylated mTOR. Finally, in the panel C, we have the PMTOR-mTOR ratio index significantly decreased in H1299 and A549 cells treated with the Bakelin compared to the control group, indicating the suppression of mTOR activator. Now I have the figure 7 panel B, and it cells expressing ASCT2 or LT1. Bakelin treatment failed to reduce PMTOR and MTOR compared to the control cells, confirming that the big cleanse effect depends on the inhibitor of these transports.
In the discussion, we compared our results with previous studies. Jan et al. Showed that the amino acid metabolism, like phenylalanine, is altered after lung cancer treatment. This supports your findings on metabolic reprogramming. Zhao et al. And Lut et al. Report that bicalines had strong anti-cancer effects in NCCLC with multiple inhibitors actions. This fully agrees with what we observed, and Xian et al. Highlighted the role of bicalines in enhancing apoptosis and reduced migration in lung cancer, which is consistent with the pro-apoptotic effects we saw. So overall, the literatures agree or results and reinforce the potential of bicaline as an anti-NCCLC agent.
Conclusions. Molecular biology made it possible to understand that genetic information is constrained in the nucleic acids and regulated complex cellular process. The human genome, although with 25,000 genes, generates great protein diversity, and its knowledge drives to genomic medicine. Genetic variation influences these predictions and drug response, paving the way for the pharmacogenomics and personalized therapies.